DNA replication is the biological process through which a cell copies its DNA to pass the genetic information to daughter cells during cell division. In prokaryotic cells like bacteria, it occurs in the cytoplasm, and in eukaryotic cells like human cells, it occurs inside the nucleus. DNA replication occurs during the S phase of the cell cycle. DNA replication begins at specific points on the DNA called origin of replication OI. An enzyme called helicase unwinds two strands of the DNA double helix and creates a Y-shaped replication fork. Single-strand binding proteins and topoisomerase enzyme attach to the separated strands to prevent them from rejoining. An enzyme called primase adds base sequence and forms primer. The primer serves as a starting point for DNA synthesis. Then DNA polymerase adds new nucleotides, A, T, G, C, to form a new complementary strand. DNA polymerase can only add nucleotides in the 5 to 3 direction. Leading strand synthesis. This strand is synthesized continuously because the replication fork moves in the same direction as DNA polymerase adds nucleotides. Lagging strand synthesis. This strand is synthesized discontinuously in small pieces called Okazaki fragments. Exonuclease enzymes remove the primers in the lagging strand. Later DNA polymerase comes again and completes the strand. All fragments are finally joined together by an enzyme called DNA ligase. DNA polymerase checks for errors and corrects mismatched nucleotides to ensure accurate replication. When replication is complete, two identical DNA molecules are formed. Each molecule has one old strand parental strand and one newly formed strand. This is called semi-conservative model.